So in the next part, we'll continue talking about cancer cells, but specifically we're going to talk about cell cycle regulation. And the reason for this is because cancer cells tend to lose control over the cell proliferation. There are some important proteins that are involved with the cell cycle, namely cyclins and cyclin-dependent kinases. These are going to be proteins, and there's a number of them that will increase and decrease in level throughout the cell cycle. And this is what are going to be signals for the cell that they should proceed to the next step in that pathway. In apoptosis, or programmed cell death, this is a form of cell suicide. When the cell has an accumulation of issues, mutagenic issues, DNA damage, whatever, it's very severely damaged DNA, and the cell can't repair it, it can trigger this process. Things like unscheduled DNA replication can also trigger this process, and we'll talk about that in just a minute. So we do know, though, that genes that regulate this process can be mutated in many, many different types of cancers, and there are many proteins involved with the apoptotic pathway, and we are still learning the order of events, what proteins are important for this process, and how they contribute to this process. So some of the proteins are going to interact with the mitochondria in these cells. You'll have two proteins called BCL2. It forms this homodimer, or two copies of itself cut together. When they're present, this will block apoptosis. BAX is a protein that's involved with apoptosis as well. When you have BCL2 and BAX heterodimer, so one copy of each of these, this is going to be an inactive complex. When you have two copies of BAX together, this is going to promote apoptosis. So depending on what concentrations you have and what ratios you have in this cell, the cell may either be more likely to commit apoptosis, or it may be more likely to stay alive. BAX is going to basically poke a hole in the mitochondria. It's going to cause cytochrome C release from the mitochondria. And the cytochrome C forms a complex with some caspases. The caspases are specialized proteases. They have a cysteine at the active site, and they cleave after aspartate residues. Their main job in the cell is to go through, chew up other proteins in the cell. Anything that has a specific sequence is going to be its target. And so basically, once they get activated, you better watch out, because they're just going to go through and chew up all sorts of problems in the cell. And this will lead to the apoptosis. Now remember, apoptosis isn't necessarily bad. If a cell is severely damaged, it might be better for the cell to commit apoptosis and perform this cell suicide than it is to remain alive with severely damaged DNA, potentially leading to a tumor. So proto-oncogenes are things that are normally involved in things like cell growth and cell division. In cancer cells, though, this activity cannot be regulated for some reason. Once they get mutated in these cancer cells, we're going to call them oncogenes instead of these proto-oncogenes. Tumor-suppressor genes normally will regulate cell cycle checkpoints or initiate apoptosis. So in normal cells, they will halt the cell cycle in response to DNA damage or other signals. This will allow the cell time to try to repair itself. In cancer cells, these become mutated and the cell is unable to respond normally. So the cell is unable to halt that cell cycle in response to DNA damage. There are approximately 400 oncogenes and tumor suppressor genes that are known, and we may discover plenty more as this cancer research will continue. Here's a list of some of the more well-studied ones. c-Myc is a transcription factor. It's also involved in apoptosis. E6 is involved with HPV infection causing cervical cancer. So it's a human papillomavirus encoded oncogene. So this is an interesting one because the virus itself will encode this gene. And it's going to inactivate a cell protein called p53, which we'll talk about in just a minute. So the virus itself is encoding this and is going to lead to the cervical cancer. Cyclins also can be mutated. They're responsible for regulating the cell cycle. These are all examples of proto-oncogenes. In terms of tumor suppressors, RB1 is going to be involved with cell cycle checkpoints, involved in many different types of cancers. P53 involved in transcription regulation, involved in many, many different types of cancers. The BRCA1, BRCA1, BRCA2 involved in DNA repair, involved in many breast cancers, ovarian cancers, and many female cancers, and as well as prostate cancers. So let's talk a little bit more about p53 in particular. This is an interesting gene because it is the most frequently mutated gene in human cancers, and in fact it's mutated in over 50% of all cancers. The way that it works is it's normally kept in check. It's bound to a protein called MDM2. This keeps that p53 unstable. MDM2 puts a little tag on this p53 called ubiquitin. This causes p53 to be turned over and unstable. However, after DNA damage, this MDM2 releases p53. p53 is stable, it's able to go to work. p53 then initiates several responses, including arresting or stopping the cell cycle so that the cell has time to fix the problems. If the problem is too large, the cell can also initiate apoptosis. So this right here is a diagram of p53. It is normally bound to MDM2 in unstressed cells. This MDM2 then causes ubiquitin to form at the end of the protein. This causes that protein to be turned over very rapidly. After DNA damage and cell stress, MDM2 is no longer bound to p53. p53 has then can accumulate into these cells, and you can get transcription of target genes, things like p21, BAX, which we talked about, important for apoptosis, GAD45, which is important for DNA repair, as well as MDM2, which is important for p53 control.
During the first phase of the cell cycle, G1, there is a chemical checkpoint that a cell must pass to begin the process that leads to cell division. The protein products of several tumor suppressor genes act at that checkpoint to control cell division. Cells with mutated forms of the tumor suppressor genes may not be able to stop the cell cycle at the G1 checkpoint, thus allowing them to grow and divide rapidly and form a tumor. One such tumor suppressor gene is the retinoblastoma, RB gene. It is often found to be mutated in a form of cancer in the eye called retinoblastoma, as well as in many other cancers. Normally, the RB protein will bind to a transcription factor called E2F. Transcription factors are proteins that can activate gene expression. The G1 checkpoint is passed when special cell cycle proteins cyclin and CDK work together to free E2F from RB. The released transcription factor then activates genes that start the cell on the path to cell division. Mutated forms of RB may not bind to E2F, allowing E2F to act without control to promote cell division. Another tumor suppressor protein that works at the G1 checkpoint is p53. This protein checks for damaged DNA, acting as a quality control for the cell. If damage is detected, then p53 both activates a DNA repair system and halts the cell cycle at the G1 checkpoint until the damage is repaired. The halt is accomplished by turning on the gene for another protein, p21, that keeps cyclin and CDK from interacting. As long as cyclin and CDK cannot form a complex, the cell cannot pass through the G1 checkpoint. The p53 gene is found to be mutated in a wide variety of cancers. In these cases, p53 fails to turn on p21 and thus does not halt the cell cycle like it should when DNA is damaged.